Welcome back to Bacteria Part 2. In this slide or presentation, I'll go through some of the other ways that we classify bacteria, and then we'll talk about antibiotics and bacterial resistance. There is one section of your notes looking kind of, the headline is Killing You Bacteria. That's a little bit out of order, so just know that you'll have to jump back to those notes at one point. And I'll mention this again at the end, but for the professional paragraph, I would like you to summarize how bacteria kind of grow to resist antibiotics, or how antibiotic resistance occurs. So last session we ended talking about the different ways in which we classify eubacteria. We can also classify bacteria based on where they get their nutrition from. So heterotrophic, which you already know, are probably familiar with that term, use food produced by other organisms. And saprophytic bacteria specifically will feed on dead or decaying organic matter, and they're pretty important to our ecosystems. Autotrophic bacteria produce their own food, and we have two different types. There are photoautotrophs, which, if you pay attention to the photo, it'll make sense that they use sunlight as a source of energy like plants. And chemoautotrophs, the chemo in there, use energy from chemical reactions to make their food. As far as reproduction, all monarans will reproduce in the same way. They produce asexually through binary fission. And because if you look over on this graphic, they are basically copying their DNA, replicating their DNA, and then splitting in two so that each cell gets one of those, one the replicate and one the original. It means that they don't actually acquire genetic diversity, but they can adapt pretty relatively easily to changing environments through mutations, especially because they replicate so quickly, and we'll talk about that later. Monarans can also be classified based on their motility. Many eubacteria are motile, and there are different ways. They'll mostly use flagella, but there are kind of different arrangements of those flagella. Now I want to look at how bacteria cause disease, especially since we're kind of looking through the lens of bacteria and human disease. They can cause disease in two different ways. So one is metabolizing the host. And in this way, heterotrophic bacteria will get nutrients they secrete enzymes that then break down organic structures and then they absorb them. So if it's in humans, they're essentially digesting and eating what's in our bodies. If the environment is in your throat or your lungs, this is kind of a big deal. And this is, for example, with tuberculosis. Tuberculosis will settle in your lungs and then it will actually use human tissue as its nutrients. In a less dangerous example, there's the Propionibacterium acnes, which causes acne. So it's probably pretty annoying, and we're probably all very familiar with it, but it lives on fatty acids in our follicles. The other way that bacteria can cause disease is through the toxins that some of them secrete. So some will secrete chemical compounds into their environment which are poisonous to eukaryotic cells or to our cells. An example of this is diphtheria, which grows in our throat. But the real danger is that the toxins attack the heart, nerve, liver, and kidneys. Another example is with food poisoning. So food poisoning will happen when bacteria have grown and produced toxins in our food, and then we eat that food. And kind of as a startling example, some of you are probably familiar with botulism, which is caused by the botulinum toxin. So 1 milligram of pure botulinum toxin is enough to kill 1 million guinea pigs. 1 milligram is a really small amount, so it doesn't take much of this toxin to basically poison someone or a guinea pig or whatever. And this is a naturally occurring toxin, and we'll talk about protecting food a little bit later. Another example, as shown in this diagram, is periodontal disease, which is in our gums, and the reaction of our bodies to the toxins of those bacteria is what causes problems. We can also kill eubacteria in several different ways. There are many chemicals that are lethal to them, but unfortunately, for example, cyanide, that's also lethal to the host as well, as you're probably already know. In this diagram there, it's just kind of highlighting some of the ways that we can attack bacteria. So we'll look at specifically cell wall synthesis, attacking them then with penicillins, and then inhibiting protein synthesis with, for example, tetracyclines. Antibiotics in general are chemicals that will inhibit the growth of just some bacteria. Two common antibiotics, as I mentioned before, are penicillin, which interferes with the cell wall synthesis, and then tetracycline, which interferes with bacterial protein synthesis. Many of these are actually derived from bacteria or fungi, and they must be in use with combination with our immune system. So they're not as, they're not very effective on their own without our bodies also putting up a fight. And then the overuse of antibiotics has led to some bacteria that are resistant to the drugs, and we will talk about antibiotic resistance as well. So again, antibiotics will work in different ways, but they will work in different ways depending on the type of bacteria. And this goes back to the gram-positive and gram-negative that I went over in the last presentation, and also that you worked on in class on Friday. So gram-positive bacteria will tend to be killed by antibiotics like penicillin and erythromycin that target the cell wall. However, gram-negative bacteria, their cell wall is protected, so they are then sensitive to streptomycin and tetracycline. And remember, tetracycline was one that inhibited protein synthesis. Then we also have broad-spectrum antibiotics, which will target either the bacteria's ability to make proteins or the bacteria's ability to replicate DNA. So these antibiotics can kind of generally work against either gram-positive or gram-negative bacteria. But whenever we use antibiotics, a few of the enemy are able to survive the drug, and over time, this can create mutations and then resistance. So mutations actually happen relatively quickly in the microbes because they reproduce so fast. And then eventually they will help the bacteria protect against the antibiotic. When antibiotics are used only as needed and as directed, they'll usually overwhelm and kill the bacteria. But as we use them more often, there are more resistant mutants arise. And when patients cut short the full course of drugs, the resistant strains have a chance to multiply and spread. I don't know if this is still a big push or if it's more kind of commonly accepted, but for a while doctors had to really push patients to take their full course of their drugs, because often you'll take drugs for antibiotics for a little while and then you feel maybe a couple days and you feel totally better, but really you should be taking them for the full 10-day course or whatever that they're prescribed. This is just kind of a representation of what that looks like for antibiotics and bacteria. And this is similar to something that you'll see in the lab that we start on Monday, looking at antibiotics and bacteria. But we're not going to be looking at resistant bacteria. So in this example, if you look at this is an antibiotic number 7. And you'll see that the bacteria, which are kind of this cloudy yellow, are totally surrounding that antibiotic, meaning that those bacteria are resistant to that antibiotic. If you compare that to this antibiotic number 6, whichever antibiotic that is, you'll see that there's this dark ring, and that's the zone of inhibition. So that's saying that it's killing off the bacteria kind of in that circumference or perimeter of the antibiotic. And then down here at number 3, that one's intermediate. So they're not yet totally resistant, but they're getting there. They aren't killed off quite as much by that antibiotic. In a minute, we'll look at some animations of antibiotic resistance. But first, in this graphic, we have our non-resistant bacteria. So there's a bacteria in your body, and it's multiplying maybe rapidly. And then you take a drug, and they all die. But when we look at drug-resistant bacteria, they multiply, you take a drug, and they continue to multiply, and the person stays sick. So let's see what that looks like.
Bacteria are becoming resistant to many of the antibiotics in our medical arsenal. One reason for the widespread resistance lies in the indiscriminate use of antibiotics. For example, at the insistence of patients, doctors often prescribe antibiotics even for the common cold. Antibiotics have no effect on viruses, and viruses cause most colds. Antibiotics attack bacteria. There are millions of bacteria in the body. Most are found in the gut and are either harmless or beneficial. Some of these bacteria are beneficial because they make vitamins that the body can use. Also, by competing for space and nutrients, the many species of bacteria prevent each other, as well as other harmful bacteria, from multiplying out of control. What happens to the resident bacteria when a person takes antibiotics? If the person has never taken this particular antibiotic before, then most of the bacteria would probably be susceptible to it. Antibiotics work by a variety of mechanisms. Tetracycline, for example, binds to ribosomes in bacteria and jams the production of proteins. From lack of protein production, a bacterium dies. Most of the bacteria die, but by chance, a few may have mutations that allow them to live in the presence of the antibiotic. Their mutations might cause their ribosomes to have a slightly different structure, which prevents the antibiotic from binding. With these mutations, the bacteria can still make proteins and survive in the presence of the antibiotic. They are resistant. The antibiotic treatment provides an example of natural selection in action. In the presence of antibiotics, susceptible bacteria die, but resistant bacteria have an advantage and multiply. The bacteria reproduce asexually and therefore make identical copies of the original cells. The populations of bacteria evolve, with the proportion of resistant bacteria greatly increased. Once established, resistance is hard to eliminate, and the resistance may spread to different species of bacteria.
So I'm going to stop that there and continue on. So this is just a chart showing some of, as they mentioned in that video, that often the common cold will be prescribed an antibiotic. But the common cold is caused by over 200 different rhinoviruses, and we know that viruses can't be controlled using antibiotics. There are several different ways where we can control bacteria in our food. One is with heat, so canning. If you seal your food in airtight jars or cans after killing the bacteria through heat, and they're sealed properly, then your food is protected. And that kills the endospores. There's pasteurization, which can, this is where you heat milk to kill harmful bacteria or heat raw milk, and also with juices and cider. And then dehydration, where you actually remove the water from the food. Bacteria can't grow when water is removed. I actually dehydrated most of my vegetable meals for my hike on the Long Trail last summer, and the food lasted my month-long hike without spoiling because there wasn't any water remaining. An example of this is uncooked noodles and cold cereal as well. Then you can also freeze the food, which will inhibit bacteria. There are helpful bacteria, so decomposers will help recycle nutrients into the soil. Bacteria grow in the stomach of a cow to break down grass and hay, and they're also used to treat sewage, organic kind of consuming organic waste, and then in foods like yogurt, cottage, and Swiss cheese are all made from bacteria that grow in milk. So that's it for today. Just a reminder for your professional summary to focus on how bacteria can become resistant to antibiotics. And don't forget to ask 2 questions that you may have, a peer may have, or that you might see on a test.